we'll start. Uh, Good afternoon, everyone. Today I'm going to deal with week 2 lectures tutorial. Let's get started. First we'll discuss with the membrane potentials, action potential propagation, and then go to some computational models, circuit models, and then uh, see about the organization of nervous system. So, uh, yeah, let's get started. So first, let's discuss about what are the two main factors for establishment of a membrane potential. So here you can see uh, here like in the first scenario, let's say this is first scenario in which you have a membrane with one side of a cell, let's say I side, the I side, and is on the left and O side is on the right, and it is separated by impermeable membrane where there is no way that particles from one side diffuse to other side, and then there is high concentration of KCl on this side is higher than KCl on the O side. Right now both the sides are having equal number of K plus and Cl minus ions, and therefore positive and negative charges are equal on each side, and therefore the electrical neutrality is maintained, and hence there is no potential difference when you measure across the membrane. This is first scenario. Now let's take a scenario where you have a membrane which is permeable to both the ions, the chloride ions and potassium ions. Since it is permeable to both and the concentration here is higher compared to here, there is a concentration gradient. Like this is on higher concentration gradient and this is on lower concentration gradient, and therefore the charges move from I side to O side, both K plus and Cl minus. But the thing here is to maintain electrical neutrality. Like why would a system want to maintain electrical neutrality? Is it is known to be the most stable state of a system. So if it is electrical neutral, it is very stable and it stays there. And since equal number of ions cross the membrane, therefore there is no charge separation and there is no charge difference across the two compartments, across this compartment and this compartment, and this leads to again a zero potential difference. There is no difference across these two. Now let's come to the third case. This is third scenario in which you have a membrane which is permeable, selectively permeable to only K plus. So this is a semi-permeable membrane which only allows K plus to pass across. Since there is higher concentration of K plus on this side compared to this side, what happens is the electrons move from higher concentration to lower concentration. So this concentration gradient uh, starts the diffusion process, and this K plus ions move to the other compartment. As K plus ions move to the other compartment, the other side, this side is losing positive charge and this side is gaining positive charge because of K plus diffusion. As this side gains positive charge, there is a repulsion among the K plus particles which are already diffused. And also you can think one more way is in the direction of electric field, the potential decreases. Therefore, there is electric field like this and there is a chemical gradient from I to O. So this process will go on until both the chemical force and the electric force both are equal. The force due to electrical difference and force due to chemical gradient. Until this point it moves, and then once it gets stabilized, at this point there is still a charge separation across the membrane, and this causes some potential difference, and that is called Vm. so which is generally called equilibrium potential. Now let's see how we can give a quantitative treatment of this. So okay, yeah, let's take a quantitative treatment of this. So let's say for any given chemical species X, in let's say it is at some particular location. Let's say this circle is some location in which we are considering some chemical species X, and for every chemical species we know that there exists a chemical potential. Let's electrochemical potential. Electrochemical potential. What does it tell is this potential tells that how easy or difficult is it to add few more species of X to this location. So how easy it is or how difficult it is depends on the sign. Like if it is positive, it is easy. If it is less positive, it is difficult. So so let's denote this potential, this chemical potential as uh, let's say for keep inside the I side, on the I side and O side. So there is a chemical potential on the I side for species here K plus. Instead of X, let's say K plus, and this is calculated as, okay, we can actually take X only. So let's take species X inside on I location, and the total chemical potential at I location is given by, there is a steady state potential. So these are all relative terms. So let's say this is steady state, which is a standard, like it's a baseline to compare some quantity with. So this is a standard potential. The total chemical potential of species X on I side is given as standard potential plus this is a chemical gradient. So this comes through Gibbs free energy. So I don't want to go deep inside that, but just say there is some because of concentration gradient there exists a potential, and then there is a electric force, which is nothing but the valence of the ion or valence of the species X plus into the Faraday's constant. There is some constant which converts, like it's like energy per coulomb of charge. So this constant does the conversion. There is a molar conversion factor, and let's say there is a some constant F, and there is some potential, electric potential at that point I in this compartment. This is the location that we are considering. It is Vi. And similarly at O side we can consider there is a potential of X at out, right? And there is a standard potential of the same species plus RT ln. Ln is natural log. Since it is on the O side, we are just mentioning it as O notation, and then electric potential at the O side. Now, what we know from this thing is always electrons move from higher to lower electrochemical potential, not just the chemical gradient. Like this is electrochemical potential. That's denoted by ECP. So this is high ECP and low ECP, and the ions tend to move from high ECP to low ECP, or whatever the species you consider, tend to move from high to low, and until when this happens is until both the concentrations are equal. Sorry, both the electrochemical potentials are equal. This slowly, like as it moves, as it moves, so I of species X is equal to I output chemical potential of X. So until this point, there is uh, movement of ions from one side to other side. And so now considering this, taking this, Now taking this uh, mu naught and this one, what happens? So mu I of x equal to mu of x. This is the where like how we get the derivation for Nernst equation. It tells how to quantify the this potential Vm that we are considering here. So here nothing but the standard potentials cancels out, and therefore only thing left is RT ln xi and ZF Vi is equal to RT ln x concentration output plus ZFV. Yeah, ZFVO. Now let's do some algebraic manipulation and bring this to this side. This results in Vi minus Vo equal to RT by ZF log of XO by XI. This is how uh, we get the Nernst potential. So for any given ion or given species across a membrane which is selective to that species, then the membrane potential is nothing but RT by ZF. Z is the valence of that, because if we are talking about ions, it is like how much deficient it is from actual number of electrons that it could have. So for suppose it is for potassium it is 1, for calcium it is 2, like that. So and XO by XI. This gives the membrane potential for this system. This is also called as equilibrium potential. So is this clear? How we got this Nernst equation?
Sir,
Yeah.
sir, you told something that higher positive means we can easily bring that ion. I didn't understand that, sir. Like chemical potential, when it is low, we can easily bring the chemical species into that location, right?
Yes. Yes. Right. So when I define it with you. Yeah. So chemical potential is how at a particular location for a given species, how easily we can bring uh, the similar species to that same location, right?
Yes, sir.
Yeah. So I think I get about it. So the lower the chemical potential, the more number of ions, more number of same species can be added to the medium, or added to that location, and therefore it's like negative is lower, positive is higher, and you're right. So any doubt here? Then
Just to confirm, sir, that's it.
okay, okay. That is about Nernst equation, and now let's extend this to a neuron environment. So what happens in a neuron environment? We'll just recap what happened. Okay. So we first asked the question: what are the two main factors for establishment of a membrane potential? So okay, yeah. So what are the two main factors that we saw is there should be asymmetric distribution of ions. That is, there should be a concentration gradient, and the membrane that is separating these two compartments should have a selective permeability. As we have seen, if it is just open to all the ions, the electrical neutrality is still maintained, and therefore there is no charge difference across the membrane, and hence uh, there is no membrane potential. So for establishment of membrane potential, we need to have these two conditions satisfied. And this is what exactly happens in the case of neuron also. So let's shift to what happens in the neuron. Okay. Before going there, let's take again three scenarios. First scenario, second, and third. So in the first scenario, there is no concentration gradient, and there is just equal diffusion of K plus ions across the membrane here. And this ion channels are selective to K plus. And then when there is a concentration gradient, there exists diffusion of higher concentration to lower concentration, and also has the K plus ions diffused to other side. This positive charge builds up an electric field in opposite direction, and there is chemical diffusion. Let's denote it by E and C. So until these two forces become equal, they come and they give a potential called this equilibrium potential, and this is determined by RTLN XO by XO. So this is the equilibrium potential that we get. Now, whereas in the case of neuron, we have a environment which is multi ion and uh, multiple uh, variable selectivity to each ion. So in this case, we can't apply this equation because this is only for particular chemical species X. But this is a mixture of species. Therefore, there are multiple ions and variable selectivity. So let's see what equation we should use to calculate here and how is that possible. And so generally in a neuron, the intracellular has higher K plus and this is internal, and extracellular has lower and higher Na plus. And there are other ions like chloride ions. There are some organic anions present inside the cell. There is chlorine here also. There are calcium ions. So this multi-ion system is nothing but a neuron. In so yeah, coming to multi-ion neurons in environment. So as I told, there are multiple ions like chloride ion, sodium ion with different concentration across extracellular and intracellular membrane. And in order to calculate this, Goldman, Hutz, and Hodgkin and Katz came up with an equation called this is a like equation to know what is the membrane potential in a multi-channel environment with different selectivity. So they came up with the equation. This is similar RT by here there is no valence term. This is Faraday's constant, and each term like each P is a permeability and ion concentration, O is out extracellular and I is intracellular. So there is a summation symbol just to make it simple, and this tells that how a system with different permeabilities, this permeability is dependent on the number of channels. Like if more number of K place channels are yeah, if higher number of K plus channels are open, uh, the permeability is high compared to if the less number of Na plus channels are open. And therefore, with permeability and different ions, this equation gives the membrane potential, and this comes out to be around minus 70 millivolts. So what happens is, like we know that K plus ion concentration is higher inside, and at the resting state, the permeability of K plus is far higher compared to Na plus or Cl minus. So K plus is far higher compared to Na plus or Cl minus. Um, Therefore, you can think as this term is negligible because of low permeability, and this is very low as Na plus is also PNA plus, so it stays near to the minus 77 millivolts is nothing but the equilibrium potential for potassium ion. Like this comes from the Nernst equation, and since there is extra redundant term here, uh, which we thought negligible, but therefore it just tends little closer to EK. Whereas if Na plus is in active state of a neuron, the PNA is higher compared to PK, and therefore then it will be closer to the sodium ion. So, so in any problem, if they ask for like different concentration and what is the resting membrane potentials, you can use this equation given if they give the permeabilities of different ions, and this is how you solve some problems related to membrane potential. So, any doubts here? Okay. Now let's see the actual mechanism. Let's understand this, how the resting membrane potential is established. So let's take the first case here in which uh, let's say the ion channels are only permeable to K plus potassium channels and potassium concentration is high inside. So initially what happens is uh, as we have seen, the until electrical and chemical forces are equal, it stays there in the equilibrium. So here, let's say it reaches the equilibrium of K plus, that is Ek. Now let's add another channel which is permeable to Na plus. That is the second scenario. In the second scenario, when you add a channel permeable to Na plus, we already have an established electric force across the membrane, like because of charge difference. This orange one represents electrical force, and since concentration gradient of Na plus is higher outside compared to inside, therefore the Na plus is driven inside. And this happens until a point where both the currents are balanced, like input Na plus current is equal to output Na plus current, and there is constant outflow and inflow of Na plus, and this I let's I denote the current, and I Na plus is equal to I K plus. At this stage, this is in this C stage. So this hole is in C and this hole is in B. So what we have to remember is from GHK equation, we see that if only K plus ions is there, the resting membrane potential, the equilibrium potential is Ek. But since Na plus channels are also there and it goes until both the currents neutral each other, and therefore it goes little higher than Ek plus. So this is how the resting membrane potential is at minus 17. And on the higher side, if only Na channels are present, it can go on. Like this would be a equilibrium potential, but that's not a case in a neuron. Uh, so this is how the electric membrane potential is established in a neuron. Is this clear? Okay. Now I have a question for you. Like, so here we see that the currents are outflowing, and there's outflow of K plus and inflow of Na plus. But uh, this if this happens continuously for prolonged period, uh, it it nullifies this, and there is no potential difference. Like slowly all charges comes up and the electrical neutrality is again established and the potential difference is zero. But for you know uh, for neuron computation purpose, we need to have some resting potential so that you can activate, and this stores the information, represents the information. So the question is neuron really at rest during resting? So uh, did you get this? Does anyone know answer for this? So it's
Voltage gating, like at that 70 potential, I think so sodium gates might get closed.
yeah, that action potential will come, but I'm talking about are neurons really at rest when during resting membrane potential? Is there energy consumption by the neuron when it maintains the equilibrium potential?
Don't have any idea.
Yeah, so what I'm saying is, since like this currents, like equal current, equal outflow of K plus and inflow of Na plus cannot continue for prolonged period, because if this continues, the charge separation gets zero, and therefore there will be no potential difference. In order to maintain this resting membrane potential, there is a mechanism in place in the neuron called sodium potassium pump. So in sodium potassium, is this visible?
Yes, sir.
Uh, so like in order to counteract that effect of continuous inflow for long period, what there is this sodium potassium pump comes into play. So what does it do is it consumes some energy from adenosine triphosphate, which is actually generated through cellular metabolism in mitochondria, and this ATP loses a phosphate ion and then binds to this. This will change the conformation. Like for every three ions, three Na plus ions going out, two K plus ions comes in, and this kind of mechanism needs energy. That is like this conversion from ATP to ADP. Therefore, like it's not completely at rest. The neuron is not completely at rest to maintain a membrane potential. It still consumes energy, and therefore it's more appropriate to call it as steady state rather than rest. So there is still energy consumption. It's not exactly at rest. So that is a point I wanted to highlight. And so when this phosphate ion goes and binds to these are some kind of proteins. So they change the shape and structure. So when the phosphate ion attaches to the sodium potassium pump, this protein, it just changes its shape such that it is open to the extracellular space and lets out the Na plus, and now K plus can come and sit. And once again the phosphate goes away, there is again like change in shape, and this process continues, maintaining the resting membrane potentials. So yes, Kirtana.
Sir, in the example that we saw, it was clear that the K plus ions were permeable and Cl minus were not permeable. That's why charge difference has come. But here in the case of neuron, both sodium and potassium are positively charged. How are we actually getting the charge like potential, sir?
Oh yeah, yeah. We are considering actually it's a electrochemical potential. So it is a mixture of forces from the chemical potential and electric potential. So that is causing, not exactly the charge of ion.
Okay, sir.
And since we don't want the currents, like as you have seen in the previous slide, we don't want the currents to be like this for long time. If it is like this, then just uh, there is continuous inflow, continuous outflow of K plus, then there is no charge separation.
Okay, sir.
And this, I just want to highlight that there is neuron is not actually in rest during resting membrane potential, and there is some activity going on. And this is sodium potassium pump, which maintains the resting membrane potential. Okay, now so let's see the equivalent circuit model of a neuron in resting. So uh, as you have seen in the lectures, we know that uh, the lipid bilayer, which is covered by, I'm just so this as we have seen, there is charge separation across the membrane, and this is like a capacitance device. So when you give a charge across, when you put a charge across this capacitor, what happens is it accumulates positive and negative, and this is also similar. So therefore, we say this lipid bilayer is equivalent to capacitor, and the potential difference is equal to a battery. And ion channels, actually, across ion channels, what happens is ions go through or go outside. So this is like a resistor. So now in the assignment, there was a question on like how does yeah, how does the length of uh, this thickness of this one and this change that is, if we have uh, in electronics, we have this thing called capacitance is given by some constant times A by D, where A is the area of the plate. And so similar to this, based on the area and the diameter, we can answer those type of questions, like by changing the length. Let's say you have a resistor or uh, some conducting wire, and if the length increases, the resistance also increases. So the analogy here is it has to travel past more obstructions when the length increases. So the resistance is actually given by some constant times L by A. So if the area of this conductor increases, there is less resistance, and if the length increases, there is more resistance. So if they ask like, what if the length of uh, ion current passage increases, what happens to the current and all, you have to use this sort of. So now let's come to the membrane potential. So we have seen, let's say the lipid bilayer is a capacitor, and then you have some, this is the sodium channels and resistor, and then you have let's denote it by GNA. So resistance, like reciprocal of resistance, is nothing but conductance, and that is denoted by G, and GNA is conductance particular to the Na plus ion channels. And you have Gk, and since equilibrium potential of Gk is negative, let's just change the electrode lengths here. So it is Ek, and there is some chloride also. So yeah, and like this, I shouldn't short this. Oh, yeah. So now we have a circuit like this. So this is nothing but equivalent circuit of a neuron, like by replacing the what? So let's take this. So what happens is during resting, how to calculate the membrane potential? Like let's say this part is inside, and this is the extracellular space. Now you have lipid bilayer, uh, and these are ion channels of sodium, potassium, and chlorine, and there is some equilibrium potential for each. So we have calculated using Goldman-Hutchin CATS equation, which tells how multi-ion, multi-selective, variable selective mediums you can calculate the equilibrium potential. But now we came to circuit equivalent. So with this circuit equivalent, where this GNA, GK, and GCl also depends on the permeability of the ion channel. So now we know from Kirchhoff's current law, if you're familiar with that, it says nothing but sum of currents coming to a point, let's say I1, I2, I3, all these three sums up to zero. Like they can't stay at one place. So whatever comes should have to flow out. So it is something like that. Therefore, here you can say, let's say INA we have, IK, and ICL. So according to Kirchhoff's current law, we have INA plus IK plus ICL equal to 0. So how would you calculate this thing? Uh, so current is nothing but conductance times, like you all know the Ohm's law, right? Uh, so V is equal to IR. Yeah, excuse me. Oh. So similar to that, we can calculate these currents like this is nothing but a potential difference across some across some component, let's say a resistor, and this is given by like G times V is I. Like, V is nothing but 1 by R. So when you sum up like this, so it is nothing but INA is equal to, let's say, uh, you have a potential of Vm here. So what happens is Ik is nothing but Vm minus ENA times the conductance of GNA. So when you sum this up like this, you get GK Vm minus EK plus GCL Vm minus EK. We want to, our interest is what is Vm. So Vm turns out to be nothing but it is conductance times that equilibrium voltage for particular ion plus G K plus E times K plus and GCL ECL by, when you just do some algebra, when you equate this to, like this should be equal to 0 and you bring Vm on one side and all other terms on other side, you get something like this and you will have sum of all conductances. So this equivalent circuit, with this equivalent circuit of a membrane, you can calculate what is the potential at the resting state. So this is at the resting state because whatever the ionic currents are going at the resting state sum up to 0, and at the resting state this is the thing, and there is no current across the capacitor, and therefore we are not considering that. Now is that clear? Is the equivalent circuit clear?
Sir, can you repeat this last point? You told current across the zero something.
Okay. So what I'm saying is, like, you have an equivalent circuit. You have an equivalent circuit, and at the resting state, like there is no current passing across the this thing. So we are only considering this. We are only considering the current passing through ion channels, and let's say this is Vm, and you have ENA, that is equilibrium potential for sodium channels, and GNA, and similarly you have GK and EK, and also for chlorine, GCl and ECl. So what I'm saying is, according to Kirchhoff's current law, whatever current, like the sum of currents at a point will be equal to 0. Like if you say I1, I2, I3, I4, the sum of all currents will be equal to 0 according to Kirchhoff's current law. So now what we have to do is, let's say there is some ionic current INA, IK and ICL. Okay, these are ions, I'm not putting charge. So according to Kirchhoff's current law, INA plus IK, like all the currents are coming to this point, let's say,
Got it, sir.
ICL is equal to 0. From this we get the equation of this one, like
Got it, sir.
GI XI, XI is nothing but particular species by sum of G. So this is the membrane potential in terms of circuit, in terms of circuit model. This is actually similar to the GHK equation, just that that is more of a chemical gradient and permeability, and this conductance G depends on the permeability of that ion. So this conductance is a function of the permeability of that particular ion, and it's almost the same equation, just that we have a circuit model, equivalent circuit model for a neuron in the resting state. And once you know the values of GK, in a problem if they give GK, GNA and they ask you to calculate membrane potential across this equivalent circuit, across the cell, you have to just use this this type of, like, you have to do KCL and do some algebra and get the things. So yeah. Now we have dealt with the resting membrane potential. Let's see how the action potential is generated and then propagated. So the thing we have is these are open channels. Right now we dealt with only open channels. So there are different types of ion channels. So the main point here is by changing the permeability of the membrane for different ions, you can actually play with the membrane voltage. So it's like has professor told that like it's like a knob. So these ion channels are like a knob, and changing the permeability for different ions, you can actually play with the equilibrium potential. So this is also evident from the GHK equation. Like you change the permeability and the resting membrane potential also changes. So what happens in an action potential is there are different types of ion channels. So uh, for now, let's take this is an ion channel which is actually initially closed, but when it sends some potential at this point, it just opens up, and uh, these are called voltage gated channels. There are also mechanical gated channels where with a force the channel tends to open. So what are these channels made up of? Like channels are nothing but there are actually a proteins. Yeah, these are proteins which are nothing but like amino acids, long chains of amino acids, and this have some charge. Based on the charge on inside and outside the cell, they respond to voltages. Some are like mechanical, like due to force they change the shape and the channel gets open. Or for example, if you take vision, the photons fall and the channel open, and then the potential rises and mechanical. So what happens is using these modulations like using voltage or force or any other kind of modulation, we are able to change the permeability across the membrane, and hence uh, the permeability changes across ions, and we are able to modulate the potential across membrane. So this is what happens during action potential. What happens is initially once the sodium channels open, the permeability of permeability to Na plus also increase. As the permeability increase, the action potential tends towards ENA. That is the equilibrium potential of system where only Na plus ions are present with channels permeable to Na plus. As it increases, the potential moves more closer towards the ENA side here. Once it reaches that point, then there are voltage gated potassium channels, which sense that once the rule they follow is once it reaches 30 millivolts, like close to 30 millivolts, they just open up, and again the potassium ions come inside. Once the potassium ions start coming inside, the potential again tends to go like permeability of K plus ion is more, and according to GHK equation, it goes more towards EK, and therefore it goes to negative. And this is how it is generated. The action potential is generated. It is mainly due to the feature that we are able to play with the permeability of ion channels, and they get modulated with different conditions. So, so there is a depolarization state. Once the sodium ion channels open, there is a depolarization stage, and permeability of Na plus increase, and it reaches a peak. Once it reaches a peak, the K plus ions open and Na plus ions close, and this comes down, and through later on to leakage channels and sodium potassium pump, again the resting membrane potential is maintained. So what we have resting state, then it reaches a threshold. Once it reaches a threshold, Na plus ions open, and it reaches a peak, and then K plus ions open, K plus ion channels open, and then it goes little down and then goes back. So this is how action potential structure. This is called depolarization phase. And since it is going, it is again getting polarized. Polarized in the sense it is having some resting membrane potential. So this is like -70, and this is called hyperpolarization phase. And this is the action potential generation. Anyone have any doubts regarding action potential generation here? Okay.
Sir,
Yes. Tell me.
sir, it gets hyperpolarized because the K plus ion gets close when it's somewhat like more than 77, right? Minus 77.
I mean, -70. If it goes below -70, it is, oh yeah, yeah. This is depolarization, and when it goes below it, so this is actually takes some time for K plus ion channels to close. So this causes more dip in the potential. So usually the K plus ion channels are slower than N plus. So this is
Okay,
K.
sir.
So this is, I mean, getting down is repolarization, and once it goes below, it is like this is hyperpolarization, and then comes back to resting stage. Yeah. Any other doubts here? So it's the same thing. Depolarization, repolarization once the potassium channels open, and since they are slow, more and more K plus ions go inside, and it creates hyperpolarization because it is like as the permeability of K plus increases, it goes more towards like -70 to -77, and then again gets back to resting membrane. Uh, these are the stages of action potential, and yeah, once it comes at this stage, again the all the ion charge separation across the membrane with the help of sodium potassium pump is again maintained. And this period, like you can't get an action potential immediately after this stage because there is some refractory period where there is some time constant to the neuron to get back to give again an action potential. And so this is one of the limitation that we have on like how frequently can you generate an action potential. So that depends on this thing. So I think last class someone were asking like what did Hodgkin and Huxley did. So I'll just cover a little bit about it. So what is the experiment? Is the figure visible? Yes.
Yes, sir.
Yeah. What did Hodgkin and Huxley did? What did Hodgkin and Huxley do? So they did some experiment called voltage clamp experiment. What does this device does is they give a command potential, like let's say I want to keep the cell at some so and so potential, say Vx, and as soon as I give a control saying that I want this potential, this circuit using some feedback, there is a feedback amplifier, it maintains the potential across the cell. So what's the advantage of this experiment is once you have a constant potential, like you maintain a Vm at this particular potential, you can see like what the ionic movements are across the membrane. So you can see how the like I'll change the command potential, how the ionic current movements are happening across the membrane. That is what you can see, and you can characterize each part of it. Like you have seen some parameters like GNA, GK, these parameters, the form of action potential, those all can be characterized, and this is what exactly Hodgkin and Huxley did. Like this is a voltage clamp experiment, and the equivalent you can understand better by an equivalent circuit. So you can actually like in the experiment what actually they plotted like how the GNA varies with changing VM. So they see something like this. I'll show them next. Yeah. So voltage clamp maintains a command potential that you give, and then you can characterize how are the currents moving across the membrane. By keeping voltage constant, you can calculate like G is nothing but I by V, right? So if you know the what's the current flowing and what is the voltage, you can do that. Like you have across this, what is the potential? Like I by Vm minus Vk. So Ik. You can characterize like by measuring the current, you can say what is the conductance of potential and how it is changing over time when you give a potential when you give command voltage of some Vm. When you give Vm, how is the G and Gk are changing? So the thing is, both the ion channels are open or closed. So it's a mixture of conductances. So how did they differentiate with Na plus currents and K plus current is they use some pharmacological tools like such that uh, like there is some chemical called TTX which closes the Na plus channels, and they can measure particularly how is Na plus varying, and similarly on using some other chemical they can or just subtract it from the other current, you will get K plus conductance. So so this is the exact like one of the graph they formed from this voltage clamp experiment. So maintain the cell voltage across the membrane voltage constant according to your command that you wish. How is the conductance of potassium and sodium are changing? They're able to plot that and characterize it, and they see like which components corresponds to the which part of action potential, and those they see like the rise of action potential is actually coincided with Na plus conductance, increase in Na plus conductance, and then the repolarization phase is characterized with increase in K plus conductance, as you can see in this graph. Like these are conductance, these are Na plus conductance, and this is action potential, this is membrane potential, and conductance is nothing but well, it's a function of permeability, and it depends on number of ion channels of a particular ion are open at that time. So this axis is given as number of open channels. So this actually characterizes the form of action potential and what particular stage of action potential corresponds to what current. So using this, they are able to characterize. So does anyone have any questions about this Horton-Huxley experiments? Last class, someone were asking like what did they exactly do. So this is what exactly they did. Something called voltage clamp experiment, where with this device they can put the membrane voltage at constant and see how the ionic currents are varying and characterize the action potential or membrane potential. Okay, is it fine? I'm moving forward. Any doubts here? Okay, now we have seen like the components of action potential and how it is being generated. Let's see how it is being so, like most of the questions deal with these things like um, so now let's take action potential propagation. So how does action potential propagation takes place across the neuron? Like this is only in one compartment we saw. Like this circuit is only one part. Like you have a neuron with some dendrite or axon at one particular point. This location is possible, but you know that neuron has an axon and dendrites, and those are like there is some kind of propagation of signal which has to be performed by the neuron. So how is that performed? So we see that once the sodium channels open, the sodium ions get in, and they actually travel across the like once they enter, they travel across the in the cellular space, and as they travel, This creates propagation of like this current when sodium current is traveling across. It also creates some potential at that point, and because charge separation is also varying, and due to that the ion channels at next location also open up, and the sodium channels are coming inside, and then the potential is increasing, and then again action potential comes. So it is like you have an action potential, you have another, so starts here and then goes here because these channels get open, and this is all. And one more thing is, like, although ions move in bidirection, why does the action potential doesn't travel bidirection? It's only traveling like down the axon, but not like two sides. Uh, so what's the answer here for that is, like, since there is an action potential already generated, so the previous location is still at refractory period where you cannot fire an action potential in that period. Therefore, even though the current travels in both the directions, the potential is not action potential is not generated in the like is generated only in forward direction, not in backward direction. So, is it clear like why the even though the sodium currents are passing bidirectionally, the potential the action potential is not generated both the sides. So for that, the reason is the previous location is still at refractory period, and therefore no action potential can be fired. Is this point clear?
Yes, sir.
Yeah. So it propagates. Now, like, what if you have a insulation at some points on this? So you have some insulation at this point and another insulation, and in between there is a gap. So what happens is the current quickly propagates, and instead of generating at every uh, sublocation of this as the current passes, what happens is it just generates at this nodes. So that way the conduction velocity also increases, and since this is insulated, there is no movement of ions across this myelin sheet, and it just jumps. So this is called saturated conduction that we saw last time, and the propagation speed improves when you have the myelination in place.
Sir, can you tell the reason for velocity of high velocity again?
I mean, uh, so since there is like high velocity, because there is jump at nodes, right? So because of that, like the current propagates, so there is no need for generating an action potential at every at every infinitesimal locations. It's like you generate here, the current propagates to the next, and like the propagation speed of current and the mechanisms of this is like compared to mechanisms of ion channels, the propagation of current is faster. So
Got it.
yeah, that's what I mean. Yeah. And so if you increase the cytoplasm diameter, what happens is the speed of current also improves. Like if you increase the diameter of this, what happens is number of collisions or obstructions that are faced by a ion is less. So suppose now we are saying about Na plus current. Therefore, if you increase if the diameter of axon is increased, the the uh, connectivity will also increase uh, for that specific species, because yeah, the resistance is low and it propagates. So yeah, okay. So now we have seen the action potential propagation. So we have seen action potential propagation. Now let's see what happens at the synaptic transmission. So uh, here, I'm just showing about EPSP. There is IPSP also. Let's talk about EPSP and then. So there is a presynaptic neuron. Let's talk about figure A here. So there is a presynaptic neuron, there is a postsynaptic neuron. This is postsynaptic and this is presynaptic neuron. So action potential reached this point. Once the action potential reaches this point, there are some channels called calcium channels. These are the calcium channels. So as the action potential reaches the end of a neuron, these calcium channels open up, which are voltage gated and calcium permeable. And this calcium ions gets inside the ion, and this creates some cascade of events at the end of a neuron, such that this helps in synaptic vesicles where uh, the neurotransmitters are stored, combined with the membrane, and just release the neurotransmitters, as you see here. And these are ligand gated channels where once this ions come and bind, the sodium channels open, and then this creates some postsynaptic potential. So this could be EPSP or IPSP depending on what type of neurotransmitter and what types of receptors are there. So if the receptor is like chloride ion channel, and this one opens up only chloride channels, then it creates like these are negative ions. As negative ions go inside, this creates something called IPSP. If these are sodium ions, this creates EPSP. So this is how the synaptic transmission goes on. So action potential comes at the end of a neuron. Calcium ion channels open, calcium gets in, which creates an environment where the synaptic vesicles containing the neurotransmitters are able to combine with the membrane and release the neurotransmitter. And depending on what type of neurotransmitter and what type of uh, ligand gated channel it is, the EPSP or IPSPs are generated. So understood the synaptic transmission? So now,
Yes, sir.
yeah, okay. This is how we have seen like action potential, synaptic transmission, action potential propagation, some equivalent circuit model, and how to calculate membrane potential and different stages of action potential. Now let's shift little bit to some computational model of a neuron. So this is, as you have listened to the lectures, this is proposed by McCulloch and Pitts. So you can think this as inputs to a neuron dendrite, and these are like whether it is EPSP, IPSP, and how strong, how many receptors are there. This weights depend on that. It's a like kind of very simplified model of a neuron, and this weighted sum of inputs pass through the neuron, like at the axon hillock, where the integration of different dendritic inputs takes place. This is like that, and then action potential is all or none phenomenon, and this step function denotes that. Like if it crosses the uh, some threshold voltage, it just spikes. And this is only MP neuron we are talking about right now, and model for this thing. And uh, how does this relate to computation is, you have seen in the lectures that we are able to model XOR gate, sorry, not XOR gates, AND gates. We can model different gates, which are logic gates. So uh, these are like depending on input, you know what output it gives. Let's say ABR input and what output it gives, and you can implement all this base, basic logic functions, and these are the same things that are in the computers also. Like you can implement logics using logic circuits which play with inputs and you get some output like that. But the thing that is missing is we generally learn like the neurons, or we also generally learn through some feedback. We have some feedback of output, like how far we are from the target output, and we just pass through that. So that creates this perceptron learning rule by Frank Rosenwald. So how to do that is nothing but you have a y, and let's say you have a target t. This is actual output, y is the actual output, and target t. The main aim of this feedback is you want to reduce the distance between these two, like the difference between these two. So the difference, let's say, is given by this thing, and we are not very particular about what sign the difference denotes. We are just what is important to us is the difference. So let's say it's squared, like, so that sign, we are removing the sign consideration. We're not considering the sign. And this is the loss function that we have. This is something called loss function. And from where did we get perceptron learning rule? Perceptron learning rule, from where did we get is, like, let's say this loss function has some function. So your initial weight is at this point. So this is, y is nothing but summation of wi xi. So the main aim of feedback and learning is to learn the appropriate weight so that you get a desired output for a given input. So how does this happen is, you want to move down the hill, like you want to minimize the loss. This is the loss function, and you want to minimize. So how do you do that is, you want to move down the slope. So slope is given by the differential of loss function with respect to weights. And as you go along this, so w plus dl by dw, this is the update rule that you have. So when you do this, you have the t minus y, let's say this is delta, and when you do this, like when you replace y by wi xi summation and do a differential with respect to each of the wi, and you get an, like this is for each, you have summation again here. So this leads to the back propagation rule, that is delta. Some, you don't want to move completely there, so you just give some scaling factor, how much you want to move, that decides how much you want to move down the slope, eta times xi. So when you differentiate this, you exactly get this delta xi, where delta is nothing but target minus actual output. Yeah, this error you back propagate and learn. So w updated is equal to w initial plus this this gradient term. So let's say this is delta wi. So you change the weights for every, like, feed forward, you get again feedback, and like that you change the weights and learn the function. This is the multi-layer perceptron learning. This is perceptron learning rule. Now we'll extend this to what is multi-layer perceptron. Is the perceptron learning rule clear? Yeah. Just the only thing you have to remember is it just goes below the slope like this, and that is how we got the update rule. And this update rule is called perceptron learning rule. So now they thought, like, by increasing the number of layers and all, what I mean by layers, like increasing the number of neurons and number of stages of this processing, it's a more complex system, and the they thought it would approximate any function of your requirement. So that leads to multi-layer perceptron. Like, you have something called hidden layers. The previous case, we only have inputs and one neuron, and you have the one output. Here there is multiple stages of multiple replicas of similar input stages. So this makes it more complex and helps in propagation. So here, how do we extend the perceptron learning rule to multi-layer perceptron? So what you do is, you have actual output and target value. This is like, this is actually a vector. Like, this could be a vector or scalar. You just take irrespective of whether it's a vector or scalar, just take norm as a loss function. So you want to minimize this loss function. So what you do is at every stage you have some weights here, weights at layer 4, weights at layer at the outset. Sorry, this is let's say 5 at the output. This is at 4. You have at 3. Yeah, you have those weights at different layers. Let's say this is a matrix. This is of size input size cross output size, and you can model this whole connections as a matrix. Number of inputs cross outputs, and for every stage you have this. For every hidden stage you have this, and what you do is you actually want to you want to learn the weights. So with respect to weights, you want to move down the slope. So similar to single layer this MP neuron, here also you can do like same thing, but what happens is since there are multiple layers, you just back propagate at every stage and you use a chain rule of differentiation instead of you know dl by. You first differentiate with respect to y and then differentiate y with respect to the weights, something like that, and at every stage you can multiply the differentials, and at, when you reach at that stage you can update those weights at that particular stage. And backpropagation learning rule is nothing but it's just the loss function differential. You're passing it to until the point of particular stage of your interest. So it's like that. So is this clear? Like, yeah, this is multi-layer perceptron, and yeah. Now let's shift again back to some neuroscience. So are these loss functions clear, or you need more explanation of this? Okay.
Excuse me. Could you please explain it a bit more?
Yeah, sure. Which one you want? This. Uh,
The perceptron from that.
okay. What I'm saying is, like, you have some input output pairs for every given xi's. You have some yi. Like for a given vector of x1, x2, x3, some values, you have a mapping from this input to some output, right? So in your data, you have some data where this xi's for each data point are very different, and for each data point you have some particular output. Let's say they belong to some class and they don't belong to some class, or if you take logic gates example, it's like 0 or 1 output, and you know for which inputs you have 0, for which inputs you have 1. So let's say you have a target t. This is what you have, and this is you get from initial, like you just initialize the weights to some random value, some random values of weights, and you pass the inputs, you get some output. But for that particular input from the data, you know what is the target output, and therefore you want to, the main aim of this is to learn that function, the function mapping from xi's to y. The main aim of this is to minimize this loss. Like here sign doesn't matter, so just take a square of this, and I'm just trying to like, yeah, you have this, and this t minus sigma wi xi is this yi. You have this, right? Now what happens? So let's say this is a loss function. This loss function has some curve. This loss function has some curve, and you want to minimize this loss. So you want to go down this slope, right? So slope of any curve is given by differential, like loss function differential with respect to like what x-axis you are following. This is like function of wi's, right? So you have function of wi, you have this loss function, and dl by dw is at any point how you move down the slope. So you want to minimize this loss function. by updating the weights, like which weight gives the minimum loss. You got it right? So when you do dl by dw, what you get is, it's nothing but like this square, right? There's a square here. So first this differential, you get uh, 2 times t minus sigma wi xi, and then you have to differentiate this with respect to wi, that gives to xi. So let's take this 2 term into some other constant and say this as eta. So the back propagation perceptron learning rule, what it says is eta times the difference by updating the weights. Let's say this is delta w. This whole thing is delta w. That is eta times the difference between target and actual output times xi. This is delta w. So you update the weight by adding it to the actual previous weight. So whatever is the previous weight vector, you add it so that you go down the slope, and slowly, like as you progress this feedback and feed forward, you reach a minimum point where the loss function is minimum, and hence you know what, like you'll get what for a given input you get the required output. That is the main aim of this. Is it clear?
Yes, yes, it is. Thank you.
Nothing so much. Yeah. Just that you want to minimize the loss, and similar thing goes with backpropagation learning rule. Just that since you have multiple layers, like multiple stages of similar processing, and you know that in differentiation, like if you are familiar with calculus, uh, you can actually use a chain rule. Like you have a function of x, and x is again a function of some u. Then what you can, like dou u by dou x can be calculated, df by dx can be calculated as df by du times du by dx, something like this. So this is a kind of chain rule. So from similar thing you get back propagation, like you multiply at each stage and come up to which stage weights you want to update. If you want to update this, you generally update all the weights, so at each stage you update. So you come with the chain rule, loss function differential with respect to first layer weights, then whatever is there, then you multiply with the previous layer output with the previous layer weights, something like that. And this is how it goes with MLP back propagation. Is this fine? Yes. Okay. We have actually seen a bit about nervous system, organization of nervous system in the. It's not very visible, but yeah. So in a brain, you have different lobes, which have different primary functions, although there is some kind of association areas and multisensory integration. But for simply, like for simple processing, these lobes are having very functional specificity, and which was taught in the lectures. In the nervous system, you have central nervous system and peripheral nervous system, which we discussed last time. So here also you can see those, and a better way for remembering the terms here is like you have this superior, inferior, anterior, and posterior, which you can see the terms here. Similarly, for spinal cord also you get the same axis, like uh, so. But since the it's a extension of brain towards this way, so there is a change of axis, but there is continuity of other terms, like ventral dorsal is not anterior posterior, but it's still ventral dorsal, as you can see. It goes like this. So you just have to remember like these terms, like what is called ventral and what is called caudal, and you have different planes that we dealt with last time. So we didn't see like how does the spinal cord is there. So what happens in spinal cord is, here you can see like uh, the white matter. So So we know that the white matter in brain is inside and uh, gray matter is on superficial layers. But this thing reverses in the case of spinal cord because more cell bodies are on the central side and therefore gray matter is inside and white matter is outside. And there are different types of cellular bodies processing different phenomenons here. So the lower side, which is nothing but dorsal side, is uh, sensory, more of sensory neurons, and the ventral side is more of motor neurons. So sensory and motor processing happens in different two locations in the spinal cord. And for example, you can see a knee reflex here. So what happens when a doctor hits near your kneecap is the basal spindle brings the signal to the sensory neuron, and then there are some interneurons. These are inhibitory interneuron. Why do we need inhibitory interneuron here is as you extend this muscle should contract. So for that you have to inhibit here and excite here, and you get a stretch then. So this is how it is organized, and you have sensory processes more happening on this side, sensory neurons, and motor processes happening this side. And this is only at the spinal cord level. As you have seen in the lectures, there are different levels of communications. That is like within the spinal cord, like just like this, and it goes through brain and spinal cord, and this can happen in two stages. One is like going into cortical area, one is subcortical area. So you have these three levels of processing. And is the anatomy of spinal cord clear? Okay.
Yes, sir.
Yeah. Yes. And you have different pathways. I think which are covered in the lectures. These are little complex, but yeah, fine. So this is somatosensory pathway. You have somatosensory cortex here, and like different segments of the small brain here, medulla and all. So whenever you touch, the sensation goes to the sensory neurons and then travels across the like medulla. Medulla it just decassate into different hemisphere and then goes propagates the signal to finally to the somatic somatosensory cortex, which processes the information as you need. Uh, any doubts in this pathway? Like, you want any specific things here? Okay. This decassation occurs because of it has some evolutionary reason that uh, when first vertebrate was formed, there is a twist in the body orientation where the spinal cord comes compared to invertebrates. Invertebrates do not have like they have same side. Like, they don't have this left going to right and right going to left. That has some evolutionary reason, and this is motor pathway. So this is a primary motor cortex. Like you make some decision to do some motor action, and from the cortical region there is a projection onto motor cortex, and from motor cortex it goes through spinal like small brain and then spinal cord, and the command is given to the muscles. So these are various pathways, and there is visual pathway. So you have like visual system is also organized bilaterally, where left hemifield projects onto right hemisphere and right hemifield projects onto left hemisphere. I mean hemifield in the sense half of the visual field that you see in real world. So before going there, like there is a very important anatomical structure called thalamus, which is like a relay station to many sensory motor signals, and then goes to subcortical regions. For example, motor system. they go to basal ganglia process which action to take, and then further passes it down. Here sensory information is like visually bilaterally organized, like left visual field, right hemisphere, right visual field, left hemisphere. And another like uh, more macro pathway is like it has two like ventral and dorsal pathway. So in ventral stream it is more of an object recognition, like what color, what form, what shape, and the dorsal pathway is more of spatial, local, and motion detection. Those kinds of stuff go into ventral stream. And yeah, this is visual system. Uh, and then you have like we have discussed a little bit about like six-layer cortical structure, uh, the laminar structure of the cortex. This is the gray matter basically. The cellular structure. These are organized based on like how based on the cytoarchitecture, how the cells are, which types of cells are there, and depending on that, the layers are classified. And you see that different layers have different inputs and like different neurons. So neurons in layer 6 have corticothalamic, like corticothalamic inputs to that, and some commissural is nothing but from other hemisphere to across hemisphere communication. And there are some connections from subcortical going to layer 5. This is in layer 6, layer 5, and each layer has inputs coming from different you know different structures of the brain. So yeah, this is about the layer structure. And yeah, so I think that's it for today. So if you have any doubts or if you need any inputs, let me know. Maybe we can discuss in the next class also. Yeah, any doubts in this lecture today's? You needed more problems, right? Like more example problems, or should I discuss the concepts?
Concepts.
Okay, I'll actually try to come up with some example problems also, because
Yeah,
sorry.
I was going to say that some example problems also would be nice.
Yeah, sure, sure. I'll get some example problems for next week also, so that I mean it will help you in the final exam actually. So I wanted to discuss some problems, but it's fine. Yeah, I'll come up with them next. Uh, so in the first lecture it was more qualitative, so I couldn't find problems, and the assignment deadline was also important. So, yeah, I think I can come up with some problems and explain. That would be better. Okay, then thank you, guys.
So,
Yeah, tell me.
sir, you have discussed a question related to capacitance and area of the plate. Like, what type of questions are related to neurons? It
Yeah,
is connected.
correct. You have a doubt in that question? How to solve it?
Yes. Yes.
Yeah, yeah. So I'll just write here. So we told uh, like capacitance like depends on the area of this. Uh, yeah. Let's say area. This plate is like a solid plate here. This is a capacitance. This is a capacitor. So there is area to this, and there is distance between the plates, right? So the capacitance of a device actually depends on like some constant times area of the plate by the difference between the plate. So here you can see that epsilon. So as the the membrane, this plate size corresponds to the area of like you have lipid bilayer, right? And this is the distance. So if the thickness increases, what happens is the distance between plates increase, and as distance increases, the capacitance decrease. So Like I'm just saying that if some problem is asked based on this, like if the membrane thickness is increased, what happens? Something like that, or area of the plate is increased, something, then this is the equation that you have to use, where capacitance is proportional to area of the capacitor plate and distance between the plate. Whereas when it comes to resistor, like there is some ions are flowing to a medium, let's say INA here, and so as the you know as the length increases, the number of obstructions also increase. So actually, the resistance is given by some constant times length of the current, that is like length of the path where the current flows by area of the conductor. It's basically like conductor is not. This is just a schema. You have a conductor with some diameter also and with some length. So if they ask how the resistance vary with increasing length, increasing the, suppose the cytoplasm is like cytoplasm, there is a, like once the current enters it flows, right? So how the like if you increase the length, the number of obstructions will increase, and you can remember that like it is proportional to length. Like resistance will increase with length, that is conductance will decrease with length, whereas with area of the the cytoplasm, the resistance decreases.
Sir, for example, like area of the uh, in the bilayer increased. For example, the capacitance has increased, then it means that the ability to store charge increased. That means the voltage across the membrane has increased. Is that the meaning?
Yeah, the amount of charge stored will increase. Like if you increase the diameter, the plate also will increase of the neuron, and therefore, like it has a more capacity to hold the charge.
That means the signal can move faster, right? Since there is more potential difference. Is it? Like the current is directly proportional to V, right?
Yeah,
Can
like this is one of the things. There are other things also, like you have ion channels on the lipid bilayer, which like with the increase in diameter, there could be density of the ion channels also could increase. So it depends on multiple factors. There is like not very direct relation. But if they ask some particular question, you have to assume that other things are constant, and yes.
okay. And sir, have one question in assignment. 1 minute. Like there is one question. Suppose you are working on a membrane potential of certain animal, and you discovered a new membrane channel that can change the membrane potential of neurons from minus 65 millivolts to minus 30 millivolts when activated. How would you characterize the function of it? I didn't get the question and
I think
options.
they're just asking like so we have seen, right, like it's like increasing potential is nothing but going towards ENA is nothing but hyperpolarization. So I think they're just asking us to characterize whether it's hyperpolarization or repolarization or depolarization.
So it is depolarization, right?
Yes.
And like I didn't understand the options were depolarization of IPSP, depolarization of EPSP. Which one of them is correct?
Oh, the options are okay. Okay. So in the question they asked, it is going towards Na plus, right? So
Yeah.
yeah, so it is like Na plus channels are being opened, so it is like EPS, excitatory postsynaptic potential, and because of it, depolarization is happening. So I think they meant that, but yeah, that options are not very clear. Yeah.
Okay, sir. Thank you. Got it.
Okay, let's meet next week. I'm ending. Any more questions? Okay, thank you. We'll discuss some example problem next week for previous, like this week's and next week's lectures also.